Welcome to the Brain on a Chip webinar. I'm Bambi Francisco Voisin. There will be 80 million people over 65 years old by 2040. That's twice as many seniors as there were in 2000. With the rise of this age group is an increase in brain disorders. It's estimated that the number of people with dementia will triple by 2050, and there is currently no cure for that neurological disease. That's why replicating the brain to find therapeutics to treat or cure these disorders has been a goal for many neuroscientists for decades. Advances in stem cell understanding and supporting technologies have helped to recreate the brain in vitro, that's outside a living organism and say inside a petri dish. But getting that environment just right is a long process. Can we get to that so-called brain on a chip in our lifetime? How are scientists and innovators addressing the need to find therapeutics that work? That's our conversation today. Welcome, panelists.
Hi, Bambi.
So I want to start with an introductory question for everyone here. Let's just start with your backgrounds and experience as it relates to drug discovery broadly. And Mark, you are on the left side of my grid here, so I'll start with you.
Sure. So I'm Mark Owusu, the CSO at Inuvio. We are a microfabrication company located in Montreal, Quebec, Canada. And my background, I've been trained in pharmacology and neuroscience, and for several years been working mostly in the ALS space. And since that time, as part of the company, we are developing tools to address the needs of the researchers in this area, not only in drug discovery, but also for basic science behind that.
Great. And Rosanna, I think you also have a lot of experience in the ALS space. That's been a focus of yours.
Yeah, that's kind of how I got started with neuroscience. So decade ago, like researching at the Harvard Stem Cell Institute, we were looking at like new pathology behind ALS and discover like protein aggregation among motor neurons. But yeah, I guess I can also go on with my background. So I'm Rosanna, VP of Corporate Development at Acro Biosystem. So we are a global leader in life sciences tools, serving over 9,000 pharmaceutical companies and academic labs across 80 countries. So and I manage the innovation and global PDNL merger acquisition company, but also started the neuroscience organoid product lines there. But I primarily based in Boston, kind of went to school at MIT for undergrad, studying computational biology and management, and then do research at Harvard, MGH, MIT in computational drug delivery design, neural stem cell, but always interested in the commercialization side. So spin-off companies in health tech and medical device. We have built a, for example, neurostimulation company to treat MCI, mild cognitive impairment, as well as depression, and have one of our lead indication approved last month. And but always kind of interested in kind of helping other entrepreneurs succeed and build up like venture capital fund with other serial entrepreneurs and invested in like dozens of companies per year over the past few years. So spending the time doing the 0 to 1, and as the kind of some of the portfolio mature, one of our LP, which is Acro, kind of asked me to help them scale as they kind of went public, and so it's an interesting journey helping a billion-dollar company scale to multi-billion-dollar company and really expanding to other adjacencies. But personally, my brother is also like has autism, so we have always been interested in really finding better cure for neurological diseases. Yeah.
Oh, thank you so much. And thank you also for calling in at 1 a.m. or whatever time it is over there.
Yeah. No worries. Always glad to chat about science and neuroscience, especially.
Hey, Jags. Share a little
Morning, and
background.
that's a hard lead to follow. That's commitment to, making other people's lives better. So, unlike my fellow panelists, I don't come from a scientific background. I'm more from the business and the commercial side. I'm an engineer who has spent about more than 20 years in the industry, seeing it from the commercial landscape, from the corporate development landscape. I've also spent some time in the R&D space, looking at the deals and the structures that are established. And I have to say that, when it comes to innovation and science, we've come a long way over the last 20 years. And, we have a long way to go, but the excitement that's there today with the introduction of AI and the speed with which we can continue to innovate energizes me and makes me get up in the morning to focus on making people's lives better. So really excited to be part of this panel and contribute to the conversation.
Thanks, Jags. And you're going to have a nice 50,000-foot view on what's happening across the industry. So we're looking forward to hearing your thoughts. So I'm going to start with just a little bit of history, looking back the last 20 years and how drug discovery has evolved, and maybe some, particularly when it comes to neurodegenerative diseases. So maybe we could talk about how it's evolved and maybe some of the top breakthrough technologies that have gotten us here. Mark, let's start with you.
Sure. So I think there's certainly one shining breakthrough technology that, in my opinion, has really come through in the neuroscience space or neurodegenerative space, and that's IPS technology. This is the induced pluripotent stem cell technology. This is the ability to reprogram any cell back into its stem cell form, if you wish, and then further from that, differentiate it into, in this case, something pertinent for neurodegeneration, which would be neurons, astrocytes, glial cells, microglia, and others. So that's really powerful because in this space, prior to that, you couldn't really reprogram, or you couldn't access any kind of tissue samples or primary in humans. So why IPS technology is so interesting for this is because now you can take human cells and start I'll use the word playing with them in the lab to get some real meaningful results and understand more about what's going on in the human brain in the end. Understandably, this is a bottom-up approach. Of course, this is a very low, high-tech level, but if you think about the complexity of an organism, this is all in vitro type work. And so this is, I think, one of the major breakthroughs in this space, which has contributed to our understanding of neurodegenerative diseases. And the second one I would say is the kind of moving into the 3D space. So this is moving that type of technology where you can reprogram cells into microtissues or what they call spheroids or organoids. And this allowed for the, making complex or advanced culture models in a dish that could be studied. And so insights that could be gained from these experiments would allow us to move forward and advance our understanding of neuroscience and neurodegenerative diseases. So I think those are the two major ones that I would say.
So the IPS, the induced pluripotent, how do you call that? And then the organoids. Stem cells, and then the organoids. And all
moving those
introduced in the last 20 years.
Yes.
Great. And what about Rosanna? Do you have something, other breakthroughs that have helped, or want to double down on what Mark just said?
Yeah, yeah. I guess I wouldn't agree more with what Mark has said with iPSC, but I guess I can add a little bit more to it. I think in I guess 10 years ago, right, we are able to do kind of iPSC differentiation into the 2D level. And I think over the past few years, we can really see it at the 3D level, and really having those brain organoid at the different kind of stages of maturity and a greater understanding of it. Like for example, when we are looking at we can gain further of even a single neuron level of EFIS data, like electrophysiology, actually getting the electrical signals, since neuron kind of communicate to each other really through the a lot of the opening of the ion channels. So that kind of gives the electric signal where we can use as another biomarker. So we can really see electrophysiology playing a greater role both in companion diagnostics as well as neurostimulation to treatment, but also when we are looking at it from like I guess today's topic of brain on a chip, we can collect those signals as a biomarker. Like we had a collaboration or a customer with Merck, right, as well as diagnostic biochip with this EFIS provider. So we were able to actually see the electrophysiology data across brain organoids that are from like 1 month of age to 2 years, and can really see by just looking at the EFIS itself, the aging of the brain. So those are some kind of impressive data we have seen. And then, of course, like we are also building kind of different disease models on top of those brain organoid, right? We can create Alzheimer, Parkinson, and kind of different disease um, state, and that really enables a better kind of drug screening in terms of drug efficacy study. And I think the iPSC route we have seen is kind of, I guess, 50%, right? Going forward, iPSC derived a disease model, and the 50% staying with animals. But because of the human brain and animal brain are quite different, I think if there is a more standard way of measuring kind of brain organoid, we'll see greater adaptation towards using that as the standard drug screening model. But there are, of course, still a lot of work in progress.
I think you talked about sort of standard way of making the organoids, and I think that's where Mark's devices come in. But I do want to ask you, you mentioned brain on a chip. Of course, this conversation is about brain on a chip, and at a high level, I want to understand if we really need to get that brain on the chip, the entire brain neural network. Do we need to map that out? And I was doing my homework, and I found that scientists have yet to map out the neural network of a mouse, which has 70 million neurons, and the human brain has 86 billion neurons. So, and 100 trillion connections. So how important is it to have this map? Does it really tell us about the function of the brain? Do we need to get, I know clearly you're already having these creating these disease models without having this map, but how important is it?
Yeah, I think there's a lot of like government initiative towards it. Like NIH has their brain initiative, which has a great focus on the brain network. And I think over the like for the past few years, as well as for the next few years, we have already mapped out a lot of those neural networks already. So I think the work now is really based on those neural network that's mapped out, how like what's the implication behind it. I think that's the state where we are in right now. And then in terms of actual its relevancy towards developing new therapeutic options, it is more like, for example, a lot of biotechs that we work with, right? They will be asking how can we tell like whether the Alzheimer disease is treated, right? Like so then we can look at the protein aggregation, for example, as a marker. So that doesn't directly involve the whole neural network activity. So there are certain key markers that you can look at that does not involve the neural network, but the industry also like to, I think it's not about neural network, but rather looking at like more data points where we have seen a rise towards multiplex. I think that's why, like as well as proteomics has been hot, as well as single cell seq, right? So I think those are some techniques people use to look at more markers at the same time, like during the early stage discovery, and then they move towards lower plex as they go to preclinical or clinical steps. Yeah.
To weigh
Mark,
in on that, if I might.
yeah.
So no, I agree with Rosanna. Except that I'd say you touched on something, Bambi, which is really important, is that, well, our understanding of how the brain works as the organ that it is, the multi-center, millions and millions of neurons, is quite poor if you think about it. And the goal here of a brain on a chip may not be necessarily to have the entire brain on a chip, because it is so complex, and you'd be inundated with everything going on at the same time, even if you could model all of that in a dish. So the idea of breaking it up more into neural network or individual systems within that brain is much more attractive and arguably more manageable from a scientific level, in order to get the result that you're interested in. And that result, in this case, might be toxicology, to assess the toxicology of some new type of molecule, or to assess mechanism of action. But coming back to what I was saying, you don't necessarily need to have all the systems in place to get that answer. And so, the technology where we're at now, we can't make a full brain, entire brain, human brain on a chip, but it may not be necessary or required, and I would argue may not be feasible. Certainly not today. But that doesn't take away from the fact that these systems and these culture advancements aren't useful. They're useful for what they're designed to be, and that's as aids or processes and assays to help us develop interesting molecules and interesting therapies and diagnostics, and identify biomarkers, which will help us in other areas. So it's really a tool, as opposed to, of course we'd like to have the whole system, but we have to think about it realistically is worth it.
Mark, I'm thinking of this metaphor that I thought of the last time we spoke, which is, if you think of the map of the United States, you don't need the entire map of the United States when you're looking for this errant car disrupting some highway. So you need to focus on that specific area and a map of that, and not the entire. That's just excessive. We don't need that. So great point on your part. I'm sorry, did you want to add something?
Yeah,
No,
I guess.
okay, sorry, Rosanna.
Oh yeah, I was also going to add like I think with even brain on a chip, what we have right now, we already are able to see a lot. Right, for the brain organoids we culture, we are able to see the 5-6 layers of the brain. We are able to see the vasculature, right, as well as the immune system embedded in it. So those are already quite valuable to simulate the actual microenvironment.
Great. So Jags, I do want to bring in Jags. I know your focus is largely on oncology, but I think that, there's a lot of things we can port and learn and bring over to other diseases. Can you draw some parallels in your world of oncology? What advances have been made in understanding how cells become more cancerous?
Absolutely. As I was sitting and listening to this conversation, I was reflecting on, okay, some of this work, if you look back, right, the genetic mapping as an example. We started this so many years ago, and now we've reached a point where we understand how these genes mutate and how they interact and how they impact the treatment, right? So it's sort of a similar stage where neuroscience is, and neuroscience has historically always been the hardest therapeutic area for the pharmaceutical companies to sort of break-in. And if you go back and look at the history, and companies like BMS had a dedicated therapeutic area focused on neuroscience. And I remember this was 2010 or 2011 where they decided to divest from it. But now they're getting back into it. So I think, as you think about it from a commercial landscape or from big pharma interest landscape, it is coming back because, the work that has been done in the research labs throughout the last 10, 15 years has evolved and reached a point where now it's piquing interest again from these big pharma. But to come back to your question on drawing the parallels in oncology, right? So if you think about oncology 15 years ago, chemotherapy was your only option until IO treatments came along. And again, if you think about IO, it is more about treating, it is about having your body fight the cancer inside and sort of stimulating your immune system to fight naturally the cancerous cells, right? And the next iteration of that came about with how do you sort of now narrow it down and make it personalized medicine, right? So they drill down on the biomarkers, trying to understand which biomarkers affect which tumor types and how the medication would either boost the immune or kill the T cells or the cancerous cells, right? So there is a lot of evolution that has happened, which I can see that the neuroscience field can learn from. It's a journey, right? In the corporate speak or in the strategy speak, we say you have to have a roadmap of vision to reality. You have to think big. So to your comment, it's about can we get the brain on a chip, right? That's our vision. Now, we may take baby steps and move towards how oncology moved and maybe focus on that road where that car is disrupting how we drive, right? Just zone in on that because that's the immediate step, immediate evolution that we can focus on. So yes, I see this story playing out again as it played out in oncology.
But with it sounds like what's new is the biomarker testing, which is essentially finding those cancer-causing genes, personalized individual cancer-causing genes inside an individual. Are we farther along in biomarker testing in oncology than we are with neuroscience? Is that an okay question? Does that make sense at all?
I would say so. I don't come from the oncology space, except the access to primary tumors means that you can identify these biomarkers in a more readily and confident way. And I think that's one of the big differences between neuroscience and oncology is that access to primary tissue. We just don't have that in neuroscience because one, if someone would give permission to take a sample of your brain, you could, well, that's the reality. You can take a sample of a tumor and it keeps on growing because the nature of a tumor, it's immortalized generally. I'm maybe making it simpler than it is, but I think it's a power where you can actually assess these biomarkers and identify them more easily. And I'm not taking away from the work that's done there. I think neuroscience has a lot to learn from this, the approach taken, and the ways to get to that will be with, and this is what I believe, will be with IPS and 3D technologies, and more towards a personalized type of thing. So we can personalize someone's cancer by cancer type. In the case of Alzheimer's, 90% or so is a sporadic disease for which the reason that it occurs is unknown. This is the same case for ALS and many other neurodegenerative diseases. Most of the cases are not genetic. So the signatures are going to be much more gray. There's nothing that's going to be necessarily standing out so much. Now we're trying very hard to figure that out. And so, but it's using the technologies where we're at in the neuroscience space compared to the oncology space. we're just a little bit behind, and it's coming. The tools are coming, so that's why it's more attractive, I think, for biotech pharma.
And I would agree with Mark's comment. You know, the advantage oncology has is the access to the tumor tissue, right? And if you go back and look time, right, oncology also started with sort of how can we, it's not the ultimate cure. They didn't start with like, let's find the root cause. It was more about how can we extend and enhance the life. It's not, I don't call it as permanently removing cancer, right, or getting rid of it. It's more about managing the disease, reducing the tumors in size, making sure they don't come back again, right? So it is an evolution, and the more they learned, the more they had access to the information, and they evolved. So I think it is a similar path that potentially neuroscience can go into, and when you look at the amount of innovation and the computing power and the speed, right? When you think about 15 years ago, it used to take almost 8 to 10 years to get a drug to the market. But when IO came in around 2010, when I go back and
IO is for
immuno-oncology treatment options, IO therapies, right? When they came with Opdivo for treatment of cancer, right after that, a couple of years after that, you will see 3 or 4 indications getting approved. So the speed of innovation accelerated, right? When they found that this was working, it went from a 10-year window to a 3-year window. Like the drug, the indications were getting approved in a 3-year window timeframe. So I think you're waiting, the innovation is happening, and I think we're waiting for that, the first success story, you know, as Mark and Rosanna are talking about, you know, the organoids. It just takes one success story, and then it just leapfrogs from there.
So, Jack, I want to get deeper into the products that are coming out from Mark's company as well as Rosanna's. But Jags, you mentioned something about BMS, Bristol Myers Squibb, getting back into neuroscience R&D. I want to understand why, and is it because there was an early, there are some successes, there's new technologies around neuroscience R&D. And then the other question would be Pfizer. I heard recently that they're dropping their neuroscience R&D, or they're tapering it back. What's happening in the big picture in terms of why is BMS getting back into it? Why is Pfizer not?
I mean, I can give you my perspective. It's been 2 years since I left BMS, and all these transactions have been post that. I can give you my perspective. I think my read on it is, and I don't know why Pfizer is getting out of it, but specific to BMS, it seems like they're looking to diversify the portfolio. They have been heavily concentrated in oncology for almost the last 12, 13 years. And this is part of the diversification strategy where they're entering, I think they just did a deal with one of the early stage neuroscience companies, and they're also doing a lot of deals in the cardiovascular space. So obviously they're seeing some potential in the innovation in the field that Mark and Rosanna are working in, right? And they're willing to bet on those innovations to see if they succeed in future. So there is a renewed interest from that company specifically. But then you can also look at the struggles that Biogen has had, right, in this space. And there's a lot of learnings from that company as well, as to how you can navigate the FDA landscape. Because what we are discussing here is more on the research side. The development is a whole other animal that you have to deal with to get the drugs to the patients, right? And FDA is evolving at the same time. For example, recently they came out with something called Project Optimus. And what it is about is finding the right dose that is acceptable by the FDA for the patients, which means you have to go through multiple dose iterations in early stage development to identify that specific dose which has the right balance of safety and efficacy, right? So again, I know we're discussing discovery here, but to get to the market, to get to the patient, it's a long road. And that's why, and specifically in neuroscience, the companies are a little hesitant. And so that's the landscape today.
Yeah, well, I think we can do an entire webinar, maybe even two all-day webinar on the FDA. But let's take a deeper dive into the products. Rosanna, one of the brands for Acro Biosystems, your company, is a Neuro, which makes the tools for all of this research for these drugs to be discovered. I believe you've said these tools are proteins, antibodies, cell lines, organoids. Are these all cells that are being manipulated to replicate a brain disorder? And can you explain what those tools are?
Yeah, so we started a new product line about 3 years ago when we started on building um, that portfolio, because as kind of Jackson you earlier mentioned, there has been that rise in pipelines in neurological diseases, and of course they will need a lot of different tools to better study it. So because Acro is a global leader in common protein, so that's kind of where it made its own name. So that's why we kind of started building proteins as our initial products in neuroscience, although we have been offering tau protein since like for the past 10 years. And so we have a portfolio of about 70 kinds of proteins. So we made those through, of course, cell engineering, right? We would re-engineer hack 293 cells as a type of mammalian cell and use it and make it express proteins, recombinant proteins. And our customers would use it for making antibodies, right? They will inject it into animals, and that will kind of through the immunization process and produce antibodies for therapeutic development, as well as looking at the protein-protein interaction, seeing if it has bind to the target proteins. And we kind of cover that for more than 10 diseases, right? Alzheimer, Parkinson, depression, as well as kind of different psychedelic diseases. And then we are, as you have the proteins, right? You also need antibodies, both as cell markers as well as to look at different diseases, right? Look at the level of tau, alpha-synuclein, which are kind of key for Parkinson AD, as well as the other kind of related biomarkers. So then we build up the antibody portfolio. But for certain targets, it may not be as easy to use a protein to screen against, right? So especially for those membrane proteins or ion channels. So that's why the cell lines will come into play. So those proteins are expressed on the membranes of those cell lines. And then like our customers, especially a lot of small molecule or other modalities, they'll use the cell line to screen for their drug efficacy. So that's how it got started. But then that's only at the in vitro level, of 2D in vitro level, to actually simulate the human physiology. We realize organoid will be the better approach or after you do the initial screening, right? If you want to go in further to understand whether the drug actually hit the target or actually have an effect, then they need the organoid. So that's kind of how we build out the organoid products. But it's also because of timing, right? When 10 years ago, right, when a lot of my peers, my lab mates at Harvard have been studying organoids, but it's not as mature of a model. But over the past few years, we have really see a lot more breakthrough, right? You see like Nature Science cover papers on brain organoids. You have also see you can group organoid with animals, or you can have organoid by itself, and really understanding how you can have a microglia and all those cell types actually exist in those organoids. So then that's make it a better or a more mature tool to study it, right? Because you need the consistency, right? You need to make sure like the organoid you grow like maybe one time is the same as the other time. Otherwise, like if they're different, then you don't know if the drug works or not, right?
Yeah.
So now it's getting to the place where it is more ready for industry adaptation. Yeah.
So Mark, I believe that in Nuvio, you make those microfluidics to create those organoids. And
We do. Yes, that's one of the products that we have.
so how do you make, and you're working hard to meet that standard that Rosanna is saying, or that need for standard reliability, reproducibility of these organoids. How do your microfluidics help do this? I know one of your customers said that you were able to, I guess, reproduce the medium or the media, the fluid that these cells exist in, but why don't you go ahead and explain?
Sure. So we produce, it complements very nicely with what a neuro is doing. Because what we need is obviously cell lines, antibodies, proteins, and recombinant proteins, and other tools that are required to do the studies. And then what we do is produce, if you want to call it, dishes or vessels, in which those tools can be put in, such that an assay can be developed, which offers a more advanced interpretation ability of the system. So, Rosanna, you touched upon this, that, you might start with a very, very, what we'll call a screen, something low down, which will identify certain interesting molecules or interactions. And then if you wanted to pursue those, you would have to move forward into using perhaps cell-based models, which with HEC-293, I believe I heard, and then further organoids to hash that out, to figure out exactly what's going on, if it really is of interest. And what we do at Nuvio is we create the environments for that to happen. So this is the vessels. So in one case that you were mentioning, Bambi, we make these relatively simple devices that allow individuals to grow spheroids and organoids so that they can be used for downstream analysis, and that could be varied applications. And at the same time, we do what we call microfluidic devices, which have features which are on the same level of size as the cells, which allow you to take that organoid, for example, and grow it out through microchannels, and then you can now look at the axons that a spheroid would produce as an example, as opposed to just looking at the entire organoid. So this allows you to get a little bit deeper into the mechanism of action, and also it allows you to think about slightly different assays that you can use at that stage of drug discovery to really work on whether this drug will work or not, before you get into thinking about all the clinical and much more downstream and much more costly, I would argue, regulatory. When you get to that stage of a drug, it costs a lot of money, a lot of time, because you're in the animals, you're in the humans. If you can figure out early on using one of these advanced models that perhaps this molecule shouldn't go forward, that's a huge bonus. Fail early, fail hard, fail quickly. That's a good thing. In fact, with the attrition rates the way they are, and they always have been, most of these drugs don't get through, and most of it is because of efficacy. So you only figure out in phase 2 of clinical trials that your drug didn't work. But if you could figure that out earlier, that you had no efficacy, or that you had a toxicology finding at the in vitro stage, at the discovery stage, by using a more advanced culture model, which is based on all those the products that Rosanna does, put them into the products that we make. It makes a nice assay, it tells you the answer. You've saved a lot of time and money, and you can move on to another molecule, another target, perhaps. So our products really complement what Rosanna's in that sense, and allow pharmaceutical companies or the customer, whoever that might be, to really get the answers they're looking for. And that's the power of all of this together.
Rosanna, how difficult, I mean, it sounds like these microfluidics are so important to this process. You have to find the right microfluidics to be part of your toolkit. How difficult has it been to find the right microfluidic devices to grow your organoids?
I think it depends on the use case, but of course, I think people use it differently. And I guess for us, there's the basic version, right? So the basic version, we can just use any cell culture plate and grow our organoid in it. So that kind of serves the basic of studying different diseases and looking at like cell viability, whether the cell dies or not, whether you are able to rescue certain disease phenotypes. And then if you want to go more advanced, right, looking at the barrier function, looking at the multi-organ systems, then that's where you need a more microfluid or like a chip system. So to actually, because you need to like create a barrier, right, and then seeing the migration of the cells. So that's where it comes more into play. So I think it really depends on what we study, but there are a lot of different partners we also work with for those functions. Of course, like what Mark is doing with his company is amazing. We also have tried different organ-on-chip players before, like tissues and methods, and like there are about, I think, about 10 to 30, I think, those kind of chip providers or instrument companies that you can test, but not everybody has a very strong cerebral expertise or brain expertise, like the liver toxicity, as well as the, I think liver and intestine systems are the more mature ones, and the brain and the cardiac one are harder. So those are not, so those are still developing. And then there's also like imaging companies like Sartorius molecular device. So they also enable the users to look more into the cells at the 3D level and giving out different information. So yeah, but it's definitely a topic that I think the whole tools industry is very focused on tackling now. From the big players to the startups, everybody is creating their own like organoid solution and trying to accelerate the process. Yeah.
It's interesting that you said everybody's trying to create their own solution. Jags is somebody who focuses on the collaborative processes. What are the challenges of getting companies to work together? What typically are some of the, I don't know, sort of the, not limitations, but what do you typically see as people hesitating to work together? Is it because they want the proprietary system, or it's just too risky to partner? What's that process like?
So yeah, great question. And I think, Mark and Rosanna are just hearing them, they're thinking along the right path as how this has evolved in oncology. But to come back to your question, it's a three-pronged approach to the process. So when you think about it from the pharma companies, what are they looking for? So they're looking for one single partner that can do or at least coordinate this work so it's less of a burden for them, right? And to Mark's earlier point, if there is a partner that can do this modeling and testing and reducing that discovery timeframe, or at least getting a decision whether there is a potential for a specific target so that they can further invest in, animal studies or tox studies, right? Because it's always the funnel, right? How can we squeeze the funnel vertically to be able to deliver for the pharma side? Because that's how they are thinking about it. Now, when you come from, the provider perspective, it's all about the platform today, right? Every biotech who is in this space of, target validation or cell line validation, right? They are looking at it from a platform perspective. All these large language models that companies are building around medicinal chemistry and biology, it's all about, can you bump off all these targets and get a quick result with high probability, medium probability, low probability, right? And the challenge from the pharma side is, well, we may not be interested in the whole gamut. Can it be sliced and diced to meet, let's say, for example, even within oncology, I'm focused on a specific cancer type or a tumor type, right? So it is sort of tailoring to what your customer wants. And as you're seeing, there's multiple players in this ecosystem, right? And end of the day, it's, can you bring all of them together to work cohesively towards a common goal? And that's the challenge, right? And all the financial incentives have to be lined up. IP has to be lined up, right? Because everybody is doing innovation and want to get compensated for that IP. So all those legal things also come into play. And I've seen in my experience, when we do the deals, whether it's a licensing, whether it's acquisition, IP is the first topic that comes up, right? If I am investing in, let's say, Mark's technology, I want to make sure that when I go to commercial, nobody is going to come and challenge that IP, because I will be potentially investing millions. And that's the last thing I want when I get to the commercial stage, somebody saying this IP is not valid. Okay? So all these intangibles, let the science alone, all these intangibles come into play when you are thinking about working in an ecosystem. Right? So those are the real challenges that exist today.
You said something about everybody having this common goal, which makes a lot of sense. If we could have everybody sort of focus on one thing. Unfortunately, with brain disorders, there's multiple. And then obviously different ways, different approaches, different tests. I looked up the first FDA approval for Alzheimer's was, I guess, 2023, if my research is correct. It's LEGEMBI, Legembi. It slows down Alzheimer's 27% over the course of 18 months. As something that has received FDA approval, is this something that the research side, this is something you would focus on and trying to help create and organize, or the devices to help study this particular drug?
So I would say, yeah, I think it's encouraging because it tells you that in that space, something was approved. So in this case, this is tau-based. So it kind of validates the idea of doing research in looking at tau as a target. So you can now, well, what is it about tau specifically, and I don't have the answer to that, but what is it about tau specifically that has to be done in order for people to live longer with Alzheimer's, et cetera? And a drug coming out would fuel the interest from a company like ours to design something specifically to look at that, tailor it, use the tools that are available, cellular tools and proteomics tools, as well as our devices and cell culture, to tailor something to further research in that area. So yeah, I do think it's encouraging. I do think it helps.
Yeah, we also kind of tried some of those drugs, as well as the Eli Lilly one, on our organoid, and seeing that you can actually see the difference in the aggregation level. So in actually rescuing some of the pathology. So I think that kind of gives kind of more hopes that we can use the system to develop better medicine. And of course, I think given some of the recent approvals in that stance, we also see more players investing in neurological diseases. So that should generally accelerate the whole process.
All right, I want to talk about wrapping this up here. If everybody could take out your crystal balls, please. And what is in the next five to 10 years, what does your industry look like, specifically for Mark and Rosanna? You'll be talking about NDD, JAGs. I'm just going to let you riff on that based on what they say. But what does the industry look like for really producing these drugs for these brain disorders, even like dementia, which has no cure? Mark, let's start with you.
I think it's going to be about the right disease model. I think we haven't yet. It might be tempting to say that genetics are everything here, but they're clearly not. So I think that the right disease model is what is the next step that's going to come out in order to use that model then to look at therapeutics. So I should back up. I don't think there's going to be a therapeutic based on these yet. It's still early for that. Of course, it could be wrong, but I think that's where we're at, is really defining what model, what assay, because you can use these genetic tools, you can use these culture systems, but they have to be tailored to the idea or tailored for the function of what you want it to do. And that's the tailoring is what I'm talking about. It's getting that assay to give you something, an answer that's robust enough that it can be used to identify or to screen molecules. So I don't think it's a therapeutic yet, but I think it's the step towards it. That's what I think 5 years for a lot of these NDVs.
Okay. Rosanna?
Yeah, I think I guess in general for our generation, kind of few people pass away because of oncology, but like neurological diseases is still like a main issue that I think we worth like tackling further. And over the next 5 to 10 years, in terms of kind of drug modality, I think to tackle neurological diseases, I think small molecule may still be a common approach because it is still difficult to penetrate the BBB, the blood-brain barrier. So we still see a lot of neurological kind of therapeutic company using that as an approach, but there will be more on the RNA side as an intervention approach. And I think AAV may solve some of the rare diseases if it was a known mutation. And cell therapy is something we are kind of still watching because I think CAR-T and CAR-NK works for oncology, and then there are a lot of startups trying to do iPSC-derived neurons as a intervention approach, but none of them really passed the phase 2, phase 3. except for approvals in Korea and Japan. But potentially iPSC derived neurons, or even there could be iPSC derived cerebral organoid as a treatment option. I think we can see anticipate more activities in that, but that one is still quite risky. But yeah, small molecule as an antibody as a treatment option, and then having the brain on chip system as a, or organoid as a screening technique should accelerate.
Thank you, Rosanna. Jags, you want to?
Yeah, I would say this has been a great session to listen to two of you, and it's been refreshing to see that there's focus on the neuroscience disease area. And look, human brain is a very complex system, right? And in my opinion, any advance towards it is a success. Call me an eternal optimist, but that's what I believe. And I think just hearing all the work that's going on, having spent last 20 some years in oncology, I was not close to the neuroscience, but I think we can make progress collectively as an ecosystem. And it's just a matter of chipping away at that next small success story and building on top of that. We just got, as you mentioned, we got an approval last year for first Alzheimer drug, right? That's a success to me. And collectively as a community, we need to build on top of that and keep chipping at it. Maybe not in our lifetime, maybe the next generation we'll take it to another step. And don't forget that computational power is exponentially increasing as we speak, and that enables a lot of innovation as well. So I think we're on the right track. Don't have a crystal ball with respect to time, but I'm very optimistic based on what I heard today.
Yeah, I mean, think about it. The first FDA approved drug for Alzheimer's, that is incredible, just last year. So yeah, and Bristol Myers Squibb starting to rethink about investing in this area. So all positive. And I just want to thank all of you. I mean, it's clearly life-saving work that you do compared to what I do. So thank you for all that, the work you're doing in the medical field. I want to mention that this will be on video. This will be also on audio. So it's up there for people to look back on, or a new audience to look at. We'll also continue this conversation on July 9th. So Brain on the Chip Part 2. So maybe you guys will be there as well. So thank you, panelists, Mark, Rosanna, Jags. Thanks to all who attended as well. Thank you for a very robust discussion. We're going to wrap it up there. And thank you again for everybody who've been listening to Advancing Toward a Brain on a Chip. I'm Bambi Francisco Roizen.